is a National Geographic Explorer? Let me explain this. Let me explain. National Geographic Explorers have obtained funding from National Geographic. And so there's two parts of National Geographic right now. There's the part that's the films and all the stuff you see on TV and all the media stuff. But then there's also the research component of National Geographic. So there's basically two separate parts. And so we are funded from the National Geographic Research Institute. I don't know exactly what you call that. The Society, the National Geographic Society. And so we are funded by that, and various opportunities exist for people to apply for funding, and they give grants and opportunities to people who are doing very interesting work, like these young people here in front of you. So what we're going to have today is a number of us, all 6 of us, are going to get up and give a little bit of a talk about what we do, and then we will also have a panel discussion. You can ask us questions afterwards. So I think it's going to be about 7 to 10 minutes for each of us, and then we'll go on. So how many of you have had National Geographic in your life? What kind of things have you done with National Geographic? You've had magazines, you've seen the TV shows, you've seen everything over 50, 100 years. I think it's from late 1800s is when National Geographic was instituted. And so again, they were bought out by, what's the guy that bought out National Geographic? Murdoch, yeah. And so basically when that happened, it divided it. And so now there's actually even more money for research, which is really helping us and the people doing the research. And we go to exotic places and we study interesting things. So I think you'll enjoy us. If you have any questions, you're welcome to stop us at any time. But I think if you, we'll have our little talks first, about 7 to 10 minutes each, and then each of us will be able to ask questions either right afterwards or as a panel afterwards. Any questions from you right now? Well, great. So please, we are going to start with Joe Cutler, Dr. Joe Cutler, who's about to go to Gabon next month to continue his research. Thank you.
Okay, I'd like to start with just a quick poll of the audience. How many of you have heard of the Ogooue River? If you have, you just raise your hand. Nice and high, proud. Okay, how many of you have heard of the Nile River? Show of hands. Okay, big difference, right? This is the Ogooue River. And more water flows out of the Ogooue than flows out of the Nile. The Ogooue is home to hippopotamus, manatee, freshwater turtle, over 400 species of fish, and every species of crocodile in Africa. The Ogooue is considered one of the most pristine large tropical rivers in the world, and it's Gabon's great river. But like many of the great rivers in the world, the Ogooue is threatened. There are 28 proposed dams in the Ogooue watershed, including 14 proposed dams on the main stem. My name is Joe Cutler, and I'm a freshwater ecologist, ichthyologist, and conservation biologist. I'm also a National Geographic Explorer, and my work has taken me around the world from Malawi to Mongolia, studying freshwater ecosystems. And I believe that the future of conservation lies in freshwaters. Freshwater ecosystems, our lakes and rivers, cover less than 1% of our Earth's surface, but they're home to over a quarter of our world's vertebrate biodiversity. And to protect something, you need to know what's out there. And so I spend a lot of my time exploring unknown rivers and figuring out what biodiversity is out there so that we can protect it. Now, my research is focused mostly in Central Africa and the nations of Cameroon and Gabon. And working in Cameroon and Gabon as someone who studies biodiversity is absolutely awesome. When you are a scientist and you study fish like I do, and you may be the first scientist to ever fish a river system, when you're pulling your net through that water and you don't know what fish you're about to catch, there's no more exciting moment. And I can tell you, I've encountered some really incredible fish over my career. Now, it's not always glorious or glamorous. I spent a lot of time, trudging through the muck and the rain and getting bit by mosquitoes and ticks and wasps and all that stuff, and catching little teeny tiny fish like this baby Hemichromis elongatus, which I'm totally psyched about. But truly speaking, over the course of my career, I've seen some amazing things. On every expedition, we have the opportunity to encounter species that are new to science. On average, about 10 to 15% of the fish species that we collect in Central Africa are unknown, unnamed, undescribed species. That's about on par as if we were surveying the bottom of the ocean. Totally cool, right? And some of the discoveries, to me, this is my favorite discovery. So it all revolves around this fish. This is an electric catfish. And this swimming sausage, they can grow up to be like 2 feet long, as thick as your forearm, and they can produce 400 volts of electricity. Now that is enough to paralyze potential prey items and to shock the unwitting ichthyologist who stumbles into the water with these things. Now everyone in sub-Saharan Africa knows these fish. It doesn't take long to learn what an electric catfish is. But no one in the world knew that there was a mimic to these electric catfishes until in 2014, my team and I collected this teeny tiny tadpole disguised as an electric catfish. Now I can assure you, this tadpole produces no electricity, but it looks so similar to the electric catfish that it wards off predators. Predators don't go after it because it looks like this other catfish. Totally cool, totally amazing. But freshwater ecosystems aren't just rich once you get below the surface. Freshwater ecosystems are like magnets for terrestrial biodiversity as well. So pretty often you'll find on the banks of the river these beautiful groups of butterflies. And if you didn't know, a group of butterflies is actually called a kaleidoscope. Fun fact, tuck that one away. I also didn't know that butterflies pee so much. So if you look at this one that's got his butt facing us and you watch it for long enough, it's gonna pee. And I never, whoa, right? Never would have known until you get that in super slow motion. And it's really easy. I mean, you're out there on the rivers, you're totally excited by the biodiversity. It's easy to get distracted. Another day you may show up and right where you're supposed to be fishing, you got a big elephant swimming in your hole. Now this is a bull forest elephant. You can see by the long, straight tusks that this is, a big boy. And forest elephants are notoriously aggressive. They like to trample you, they like to gore you, and they don't like you fishing where they like to bathe. So I'm always, a little freaked out. I love elephants, but I don't love sharing my fishing holes with them. But you also share freshwater ecosystems with some even more frightening biodiversity. So for those of you who are squeamish or faint-hearted or anything, close your eyes, look away, because this next slide is going to show a chingo flea. Now this is the world's smallest flea species, and it's a parasite. And what they do is they burrow in your skin, usually on your feet and or toes, and then you've got to surgically remove them. So you cut a little hole in it, and you pull them out, and you yank on them. And I've had so many of these things. No way. At one point, I had 8 in 1 foot. Yeah, that's my foot. And actually, she's the one cutting it out. That's a trooper. And so you may wonder, why would I do this? Why would I keep going to these areas where I've got to compete for space on the river with elephants and parasites? And my response is, these dams are being built, whether I'm out there or not. And if I'm not out there, if my team's not out there, dams are going to be built before we've ever surveyed these rivers. And that means that we could lose species. Species could be driven extinct before we even know they exist. And so I'm going out there to fill in the gaps in our knowledge, to figure out what's out in these sites so that we can actually fight for their conservation. Which brings me back to my first question. How many of you have heard of the Ogooue River? My next project, my next National Geographic funded project, is a project called the Ogooue Megatrans Site, where we are going to survey Gabon's Great River from the Congo border all the way to the Atlantic Ocean. 1,000 kilometers, a 6-month journey in inflatable canoes and rafts. And we're going to be surveying the 4th largest river in Africa. One of Africa's last wild rivers. If you're interested in following along, we'll be out there for almost the remainder of 2020. So we're on Instagram, and we love for you all to follow along with our journey. Thank you so much. our next speaker is Julia DeMarines, and I promise you it will be out of this world.
All right, thanks, Joe. I'm going to move this so I can see the... All right, so my name is Julia DeMarinis. It rhymes with Venus. And I was a 2018 National Geographic Grosvenor Teacher Fellow, a 2019 National Geographic Explorer, and most of us here are part of the 2020 National Geographic California Explorer Chapter Hub Leadership Team. And I study space, outer space. And so space, I don't know if you know this, but space is really, 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 really big. So I'm sure there's a lot of brainiacs in the audience. I can kind of just feel it. I'm going to ask you all, what do you think these dots are? Nailed it. Usually I get a few stars. Those are stars. Planets. So I want to ask you all, how many galaxies are in our universe? Take a guess. Any guess?
12.
It's in the billions, and it's more than 12.
100 billion.
More than 100 billion.
500 billion.
Less than that, but very close.
425 billion.
About 400 billion galaxies in our universe. Guess how many stars are in each one of those galaxies on average? A what?
1 billion.
More than a billion. Less than a trillion.
400 billion.
About 400 billion. About the same number. So think of how many freaking stars there are in our universe. And what's the kind of fundamental question that we wonder when we look up at the stars?
Is there life out there?
Are we alone? Is there E.T.? Are we alone? So our planet is often, you can't see this because the contrast is not so great. But if you, yeah, okay, you see this little thing? This is the Voyager picture of the Earth. This is when Voyager was way, way beyond Saturn and turned around and took a photo of our planet. If you're familiar with the Carl Sagan pale blue dot monologue, this is the inspiration for that. So it's a very small 1 pixel. Our whole planet fits on 1 pixel. It's often referred to as a mote of dust floating in a sunbeam. And that one speck of that 1 pixel represents the only life that we know in the entire universe. And astronomers, we use this picture and our planet as a proxy for looking for exoplanets. So we all know what exoplanets are, planets orbiting around other stars. And right now we found about 4,000 exoplanets. And I am currently a radio astronomer, but I went too fast. And we don't have the ability to send a spacecraft out there with our radio antennas and turn back and analyze our planet. We don't have this pale blue dot in the radio frequencies. So the project that I'm currently working on with the Breakthrough Listen group at SETI Berkeley is the Moon Bounce Project, where we're pointing our telescopes at the moon. Radio leakage is emitted from the Earth, and the moon is kind of like a nice mirror. It reflects radio frequencies back at us. So here's a better visual. Here's our not-to-scale Earth and the moon. And we have these beautiful towers, like, is it called the Sutro Tower? Is that what it's called? All right, cool. I'm East Bay, all right, sorry. The Sutro Tower, it's emitting horizontally. It's not really emitting up. So those horizontal radios will bounce off the moon into our telescopes. And then that makes me happy because that's the research I'm doing. Because I want to know, what is our techno-signature? What do we sound like to an ET? And just as I was mentioning, I'm part of the Breakthrough Listen group at Berkeley SETI. And it's the world's most comprehensive SETI search to date. SETI stands for the Search for Extraterrestrial Intelligence. And right now, these are all of the telescopes that we work with or are in partnership with to scan the cosmos or any anomalies that cannot be explained by natural phenomena. So part of the work that I do is inspiring the next generation. Because when I was 13, I saw the movie Contact, and I saw somebody that looks like me, doing really cool research, trying to find life in the universe scientifically, not some like crop circle UFO stuff. Hope it didn't offend anyone. And I was like, I want to be like this. And I was hoping to give that experience back to the next generation. So through National Geographic and some colleagues, we formed the Ad Astra Academy, where we inspire underserved students to discover opportunities through science and exploration. And we use science, and specifically space science like astrobiology and Mars and things like SETI, to really be a hook to drive students who want to explore. So far we've gone to Rio de Janeiro, we've gone to Bangladesh, and we worked at an all-girls school in a very rural part of Cox's Bazar, pretty close to the Rohingya camps, if you're familiar with that. Just some great photos of our students who were incredible. And most recently in November, we went to Nigeria to work with students in Lagos and in Port Harcourt to do these hands-on, inquiry-based, project-based learning to really inspire them to see the scientists within themselves so they can go on and choose the career path that suits them best, knowing that they have the tools to be an explorer, to be a scientist, to think about the world critically. And this is just a sweet slow-mo walk-away shot. We were doing a scale walk of the solar system, which was super fun. But you can't tell it here. But it was, I promise it was so fun. So that's all I have for you today. I do an outreach event called Space in Your Face. Twitter, Life in Space, and Instagram, Mote of Dust. And we're not doing questions, but I just like to think, Canopy, we're going to do questions at the end. And I'll pass them on. Thank you. I brought some stickers with me, so find me after if you want one. That means you have to stay till the end.
Hi everyone, I'm Mel. I'm super excited to be here. This is a really beautiful video of manta rays swimming, so just close your eyes for a moment and imagine that. It's not working. Okay. I'm going to tell you about my work. I'm a PhD candidate at UC Santa Cruz. We are on strike. I can talk to you more about that later. But I want to tell you first about how I got to where I am now. I have a little bit of a weird background. I worked in media as an environmental journalist in New York for a few years before starting my PhD. This is a humble brag photo of me with my idol. And I started covering a lot of wildlife crime and wildlife conservation issues. And there was a moment when I just realized that, you know what, I actually want to do conservation and not write about it. So now I'm in actually the same lab that Joe just got his PhD from. He left us all alone at Santa Cruz, and I work on a group of species that is really incredible. I'm going to tell you a little bit more about, which is the giant oceanic manta ray and its cousins, the devil rays. These animals are super incredible. They are very large, and I don't think that people actually realize how large they are. They can reach the weight of a Honda Civic and the width, wingtip to wingtip, that a giraffe is tall. So they're freaking big. I was actually diving in Ecuador last summer, and we were down at the bottom of the ocean, and I was like looking at some tiny little fish, enjoying myself, and all of a sudden everything around me began to grow dark. And I sort of freaked out. I'm a nervous diver, which is crazy for a marine biologist. But, you know, it wasn't until I looked up that I realized that it was one of these mantas, one of the biggest ones I'd ever seen, casting an eclipse over us. And all of my dive buddies were sort of like, whoa. So they're really big, and they have quite a large, much more like whale-like impact on people. The question that I always get, which I want to answer beforehand, is did this kill Steve Irwin? No. They are inspired for the sound. Can you change the audio? Okay. It's jarring.
I hope so.
Oh no. Okay. They are gentle giants. Don't look at my cool drum. What is it? Okay, they're gentle giants. This is the cephalic lobe unfurling, and this is how it eats. You can see it's about to funnel in krill into its mouth over its gills. Like any other fish, it's using gills. And this is just to show you that it doesn't bite anything, it doesn't have sharp teeth. It's actually a filter feeder, just more like a blue whale. And it's using this flying motion. So they're really unique, evolutionarily unique animals, and very gentle. They love divers, bubbles, they're really curious, this species especially. So don't think that they're murderous like stingrays. They also have some really fascinating behaviors. This is a species that we work with in Baja in Mexico, which is known for these incredible jumping behaviors. And we have no idea why they do this. I have a lot of guesses, which I can talk about, but it is a really ripe for a PhD. So if anybody's interested in that, I will help you think about it. They also form these amazing aggregations. This is a small aggregation that we filmed with a drone, and we're trying to figure out maybe why they do this. They can make these groups up to 10,000, 20,000 at a time. It's really spectacular to see. But this is one of the biggest problems for manta rays and devil rays. They're closely related cousins, because when they form these large groups, they are extremely vulnerable to fisheries impacts. So this is my little schematic of bycatch. I study essentially bycatch of these animals, and bycatch is any time a fishery or fishing gear captures and essentially kills anything that is not a target species. So say you're fishing for cod and you accidentally catch a sea turtle. That's bycatch. But for the past 20 or so years, bycatch research and mitigation has focused on these really charismatic species like sea turtles, like dolphins. If you've ever seen a dolphin-safe tuna label, we really have overlooked species like sharks and rays that are extremely vulnerable. So the system that I study this in is in the global tuna fishery, the global tuna purseine fishery. These fisheries are enormous. They cover 91% of the ocean surface, and it's where we get most of our canned and fresh tuna, so tuna for sashimi or sushi, but also canned tuna. These nets are incredibly large as well. They can reach the equivalent of 16 football fields across. So you can imagine if you're a manta ray, even if you're the size of a Honda Civic, you're still going to get scooped up. So we're working with these fisheries to study these animals and to try to develop conservation solutions. This is an animal that we work with in a small-scale fishery in Mexico. And this, importantly, is the type of animal that makes up the bulk of my work. So what I do is I work with the actual tuna fishermen and the fisheries observers who go aboard their vessel to study the animals that they're bycatching. So we're essentially using bycatch as a sampling method. So what they do for me is very kind. They take a very small clip of the tail of each animal. It's hard to see here, but this is the tail. He's supposed to be wearing gloves, but that's okay. And they put it in a little vial, and then they send it back to me in the lab. And from that information, we can start to map out how this bycatch looks. So most of my work is in the Eastern Tropical Pacific, based in Ecuador. And we can see, okay, where are we catching a lot of these animals? They have a really hard time distinguishing the different species, so we have to use genetics. Where are we catching them? Some of it is really concentrated around the Galapagos, some of it's way out in the high seas, and some of it is really coastal. And with this information, we can get a whole lot of information about the populations. I want to just give a little information though about how we actually do that. This is extremely stressful for me. This is the product of like 4 weeks of work. There are 82 manta ray genomes in that little vial. If you drop that, it's over. So I'm using genetic techniques. It's a fractional genome technology. Basically, we take a whole genome and we cut it up into little pieces that are easier to deal with. And from that, we can start to understand a whole lot of things. Not only whether these populations, what size they are and where they're found, but we can also start to infer which populations need conservation first, especially in a place where we have a lot of limited conservation dollars. So that's most of the work that I'm doing for my dissertation. But I do want to talk about one other project that we've just begun. It's super exciting. What I'm doing with these fishermen is kind of training them to take samples and training them to do all these things, but we really thought that the fishermen were the people who knew best how to solve this problem. They're spending all their time at sea with the animals, with the tuna. So what we did was set up a system where we can run mini contests with the skippers who essentially own the vessels and their crew and observers. And we're having them come to these big weekend-long workshops where we say, okay, you're going to design your bycatch mitigation technology. Whether that's somebody has designed a manta stretcher to remove the animal from the vessel, or that's designing a way to actually avoid capture in the first place, which would be really great. And we have a reward that we're going to do for that. So the first workshop is this summer. Hopefully I can come back with something fun, but stay tuned. And I would be remiss if I didn't plug our upcoming Giving Day. This work is really fun, but it's also really, really expensive to splice up a manta ray genome. So on April 22, mark your calendars. You can do it right now. You can take out your phone. That's totally fine. Just kidding. We're running a really big pledge drive at Santa Cruz, and you have the opportunity, if you want, to adopt a manta ray. I can tell you where your manta ray was found, what species it was, and send you some photos of him. Or, yeah. Finally, thank you all so much. It's super cool to talk about this work with you all. Please feel free to get in touch, and and thanks again. And next up, we have Ravinder Saigal.
Thank you, Melissa. So yeah, I am a National Geographic Explorer. My name's Ravinder Saigal, but I'm also a professor at San Francisco State University, which is in the western part of the city. You may not have been there, but you're welcome to visit anytime. It's one of the biggest universities around here. It's probably about 35,000 students. And so we have a lot of interesting research going on, and some of the students right here in the front rows are also from San Francisco State, and they can talk about their research. My research is fascinating. So I do work also in Africa. Not to say that theirs isn't, but this is also really interesting and important work. So imagine the coronavirus right now. Where did that come from? It came from bats, right? It came from bats that were living somewhere in China, and there was human interaction with these bats. This is kind of what I study. I study where do these diseases come from? And so what my interest is how does deforestation lead to emerging diseases? And so think about HIV/AIDS. That came from a chimpanzee in Cameroon. Imagine that somebody was hunting a chimpanzee and somehow got blood inside of them, and then that started a pandemic of HIV all over the world. That person started to spread it to other people in Kinshasa and Congo. Imagine Ebola. Ebola came from bats in West Africa. And it started to spread because, again, people come into contact with these animals much more often than they used to. There's a larger population of humans around, and there's less forests around. We're losing about 5% of our forests every year in Africa. That's a lot of forests. In 20 years, there will be no more rainforest left. 5% a year. So think about that. And think about how that will influence emerging diseases. That's what I'm very interested in studying. So next slide is going to show you what's happening with the palm oil industry. Who's eating palm oil here today? Probably almost all of you. It's in almost everything that you buy these days is palm oil. And so this you can see is how palm oil is increasing in all over the world. You have palm oil increasing in Indonesia especially, but also in Malaysia, South America, and also in Africa. And so the next slide I will show you is where I work mostly in Cameroon. This is Gabon, where Joe is working in this country right here. And so he also works in Cameroon. So what we do is we go to places and study their diseases. I'm mostly an expert on diseases of birds. So I study malaria in birds. So you didn't think about this, but you know about human malaria, but birds also get malaria, and it's actually a very dangerous disease for birds. Who's been to Hawaii here? Anybody been to Hawaii? Next slide. And so in Hawaii, the birds that are on the islands are not endemic birds, meaning that they are not the native Hawaiian birds. Those all died from avian malaria. So there was this boat that came over in 1826, it was called the Wellington. This boat comes over from Mexico, dumps their water in Maui, and along with that is some mosquito larvae. There were never any mosquitoes in Hawaii before that. Imagine. And so then, these mosquitoes, later on, you start to get malaria, right? They start to kill all the birds. So the birds that are left now are on the tops of the mountains, like Mauna Kea. It's too cold up there for the mosquitoes, right? And so those mosquitoes are slowly going up the mountain now with global climate change. So that's the problem. So what we're studying is avian malaria in Africa. This is a mosquito biting an apapame bird. This bird went extinct in 2004, the Ogooue. So very, very tragic situation there, especially with global climate change. What's going to happen is we'll lose these birds. Thanks. So this is my team in Cameroon. These are graduate students that work with me, and we have a fantastic team doing work about deforestation, how that affects the diseases next. So we get into some rough conditions. We work in the field 3 weeks, 4 times a year. These people are out there 4 times a year, 3 weeks each time. And last time we were there, we got stuck with flooded, and we had to climb over a tree to get out of our stuff out. Not easy work, as Joe can also tell you. We catch the birds with these nets. They're called mist nets. The birds fly into them, and they don't see these nets. And then we just take them out, take a drop of their blood, and then release the bird. We put a band on them also so we can see if the birds have been previously caught. So that's what we do. We're studying the malaria in these places in Africa. Next one. And so if you start this video, you'll get an explanation of what we're doing.
So here you can see the edge of the forest. On the right side here, this will all be gone and converted to palm oil plantations. On the left side, for example, this tree here, it will survive. This is the demarcation marked by these red lines on these trees for the edge of the forest. With this netting, catching birds right on the edge of this demarcation. So in the future, after the palm oil plantation is established, we will come here again and capture mosquitoes and birds and identify the different types of diseases after deforestation. Especially at this edge. We will look at this tree again in about a year's time at the exact same spot and see what happens to the biodiversity of birds, mosquitoes, and parasitic diseases like malaria with deforestation.
Yeah, so this was previous to the deforestation. You'll next see the picture of what happened. So yeah, they cut down all the rainforest next, and you'll see they start to plant these palm trees next, and they fill up these spaces with these palm trees so that you can have Doritos. And here's the forest remaining in the back next. So what we're finding is malaria is infecting about 30% of the birds. Our preliminary results suggest that these are emerging diseases. These birds have never seen these types of malaria before. These are generalist types of malaria that are getting into birds that have never seen it before. So it can be akin to what happened with the Hawaiian birds. They've never seen this type of malaria before, so they might start dying. This is what is emerging disease. It's a disease that people have never seen before. Any type of animal that is exposed to a new disease is called an emerging disease. Next one. What's also very important is we're studying the mosquitoes. And so we're finding that as soon as you cut down the rainforest, new mosquitoes come in. That is Aedes and also Anopheles. And so Aedes aegypti and albopictus come in right after deforestation, a few months later, those spread dengue, yellow fever, and Zika. Okay, so as soon as we cut down those rainforests, we're exposing people to diseases that are very dangerous. Next. And this is because when you have the rainforest, there's little containers around that are remnants of the people that are working there, and that can lead to more of these mosquitoes breeding. The mosquitoes love to breed in this kind of thing. They hate these mosquitoes. So that's a little bit about what I do in Cameroon. Don't buy palm oil. That's all I can say. This is my main message, that if you buy any of these kind of things, look at what you buy from Trader Joe's or from whatever store you go to and see if there's palm oil in it, and stop buying that stuff. Nutella, right up there. Mainly palm oil. It's made of palm oil. So think about that before you eat the stuff, before you put the lipstick on, before you use those biofuels. They're full of palm oil, leading to emerging diseases, loss of biodiversity, all kinds of problems. In Indonesia, orangutans, they were losing a lot of different species because of palm oil. Thank you. Next we have Diego Monteleone.
Hello everyone, I'm Diego. I am originally from Mexico City, but I've been in the US now for quite a bit, and now the Bay Area is home. And currently, I got my PhD from UC Berkeley, and afterwards I started a company that worked on building data infrastructure around sustainable energy in the West and South, and environmental justice. And more recently I transitioned to being the data lead for East Bay Community Energy, which is the largest provider of sustainable energy in the East Bay. So I'm doing that, but I'll tell you guys about this continuous passion project of environmental justice that I'm working on for National Geographic. Before we begin, I'll start with the poll. Can any of you tell me what environmental justice is? If any of you know what it is.
Environmental justice is the fact that environmental hazards affect negatively people of color and people of low socioeconomic status more than people who are rich and white.
Wonderful. That is the exact description. And just to poll, how is it different environmental justice in the US than it could be in other places around the world, for example, Latin America? Yeah?
I'm guessing it's just as bad everywhere else.
It is, but the difference in other places around the world is in the US and some Western countries. You're not really affected if you do research about environmental justice, if you do science, or if you do research about the human element that's related to environmental justice. In Latin America is the region around the world where most murders of activists happen for doing particularly that research. So touching those elements, gathering that data, being part of those communities is just itself dangerous, which is important, like some elements to bring out. So I'll tell you now about the project I'm working on. So before I begin, I'll start, just keep going. This is a video from a community in Baja California Sur, they were just telling us about that in 2014 had one of the largest hurricanes that devastated many fishing communities around the state. And one of these communities in San Buto and San Carlos is a community of fishermen who actually didn't own the land that they were living in. They were just illegally occupying it. So the army and the government moved them around to another very cheap piece of land. And this cheap piece of land happened to be, of course, next to the largest thermoelectric plants in the state. Not only that, but it's also next to the landfill that's occupied of being where garbage is usually burned by the 5 largest towns in Baja California Sur. So this is where the community, I'm coming back from the community, this is how the landfill looks like. It burns almost on a weekly basis and the thermoelectric plants are there. And this is emblematic of what's happening to hundreds of thousands of communities and millions of people around the world. People who are being displaced because of climate events and extreme weather events that really they contributed very little to, and then where they're displaced is a piece of land where they're being impacted the most while contributing very little to it. So the question is here, so what can be done about many of these issues? And this is where traveling, talking to people, being part of the country, going to these communities, as someone who works on technology and data, I try to understand what little piece I could work on putting these stories on the map, putting these communities on the map, and developing collaborations where data could become actionable to start a change. So it's going to move forward a little bit, maybe. Getting there.
Just probably
Right, so I'll tell you to start the National Geographic project. So it'll go to a map eventually.
Sorry, I may have restarted it.
Alright, so I'll just keep telling you and eventually we'll get, there we go.
We're on the map.
There we go, we'll go to the map. So when you think about data in parts of the world where there is no data, you have to create the data from scratch. And if we're talking about communities and we're talking about all these issues, we have to do bottom-up data creation. So we do three different things. Is the video running? Yes, it is, right? It should, yeah, there it is running, yeah. So we do three things. One of them is actually do community science. So these red dots that you see popping up are collaborations that we've established with nonprofit and grassroots organizations in different parts of Mexico as a pilot project where these grassroots boards engage the communities that are being affected for being a chatbot on Facebook, that everyone has Facebook in these places, for documenting pollution in rivers, big slaughterhouses that are polluting watersheds, big fertilizer corporations that are decimating bee populations in these communities, and actually taking pictures, taking videos, and popping up on the map. The next stage is gathering news feeds, live news feeds from across the region that are continuously telling us in real time environmental justice issues are popping up all the way from Colombia, Peru, Central America, and Mexico, which are the places that we've covered. So it's really bottom up, community science feeds, it's like medium level news aggregations. And the next third piece is actual raw data that's important to understand what is the environmental harm infrastructure that's happening. So collecting data in some of this country, this data is really a pain in the ass. All this data is energy data infrastructure, potentials for oil and gas and fracking potentials in many of the Amazon forests, in the seas that we all enjoy and all these beautiful creatures live. They're currently not mapped and doing for analysis. All these red maps, mining potentials and current mines exist in the country. But on top of this, like social harm infrastructure, like things that we actually all depend on, we map social demographic data. The presence of indigenous communities in purple, and then vulnerable populations regarding income. As you start layering all this data, it starts getting messy, right? So once you have all this data, you aggregate it, the important thing is to put the analytics behind it and build a hotspot of environmental justice so you can go very neatly into some of the highest resolution demographics that you can get into to start making a map for Latin America of the regions that we can start predicting could be issues of potential hotspot of environmental justice harm 5 years from now, and some of them they're actually happening right now. So in the US, for example, the Bay Area, we have a tool called EnviroScreen, which is one of the first tools that the US has for monitoring environmental justice in the US. So imagine if these data, or the Bay Area that supposedly has all the technology and data for making this actionable, is also just beginning to do it. You can imagine that Latin America is about 10, 20 years away from actually putting this on the map. So I didn't want to wait for us to doing all that, so we began doing it a couple of years ago. And now we're on our way to releasing the first map spanning Mexico, Central America, Colombia, and Peru in a couple of months. The next question is, okay, you have all this wonderful data. How does this become actionable? So I'll tell you two examples of what's happening. Sambulto and San Carlos is the community that I just keep talking. Can you hear me?
Yeah.
I have another one.
Sorry. The community I was just talking about is right here. And over there I went with... the partnership over there is with an NGO called Cerca. And Cerca is a very new nonprofit that is monitoring air pollution in some of the biggest cities in Baja California. And we traveled there, and through the data that we have been collected, we identified that this community is one of the focal points that we should focus as a pilot for piloting this technology. And now, with this collaboration, with the data that we're collecting, there's an active lawsuit against the 2 thermoelectric plants in partnership with Mexico's Agency for Public Health. So that's one thing. This is not the data doing it. This is the people on the ground actually being partaking using the data for action. Another one is actually with some friends from Chile who are also National Geographic Explorers. They're using the data of all the dams that we've mapped across Chile and all the rivers, and they are river rafters conservation. So we're using this map to create a map of festivals around many of these rivers, and also for organizing protests to either slow down or fully try to shut down some of the dams. So these are grassroots participation efforts, which are useful. We're also conversations with UNDP, UNEP, Interpol, the Guardians Initiative for Environmental Justice, to understand how can this data can be used more broadly for policies and lawmaking around environmental justice, and developing best practices for monitoring data and having this translate to laws. So this is what I work on, and I'm to your questions.
Hello. It's so exciting seeing so many people who are here who are interested in science and nature and learning about our world. So I have a question. How many people here in the last, like, say, week or so got out and connected in some way with nature? So, like, you went hiking or GART, pretty much everybody, right? So yeah. Or, you know, like me, went river rafting and pretty much got thrown out of a whitewater raft and almost died and got lost and we're connected, right? Thank you for not letting me drown. I appreciate it. But if you're also anything like me, you loved every moment that you were outside. And you tell your friends about it, and you invite them to come with you, and you want to conserve those wild places that you were in because you want to share it with everyone else, and you want everyone else to love it and appreciate it as much as you do, right? Because you're lucky that you get to be out there. Because just imagine for a second that I said to you that for the next week you're not allowed to go outside at all. Like, how would you feel about that? Right? Or what about if for the next month you couldn't get out of your bed? Double, yeah. What if for the next year you had to stay inside of your house? Like, what would that do to you? And I'm not talking about, like, just physically, like not getting exercise or not getting enough sunshine. Like, how would that affect you emotionally and mentally? Right? Because I know for me, when I go outside, that's what makes me feel good. It makes me happy, right? There's like this visceral feeling that you get when you are out there and you feel the wind in your hair and you know, the mud is squelching between your toes and you're soaking up the sunshine and you're maybe drinking up some river water. It's visceral, right? You know what I'm talking about? But we love it because that's what makes us, you know, humans living on this planet. We're lucky. And I know that I'm really lucky. My name is Kathy Ho, and I am a National Geographic Grosvenor Teacher Fellow. I'm an explorer, and I'm a certified educator. But most importantly, I am a teacher. So this is my classroom. So I work in a hospital school at the Lucille Packard Children's Hospital at Stanford University. So the majority of my kids are patients at the hospital who are there for acute, chronic, and terminal illnesses, which means they're basically stuck in the hospital. Like, they can't go outside. And some of them are literally in isolation in their rooms. So they don't have the ability to go outside even if they wanted to. So that leads us to my National Geographic project, which was